Good morning, everyone. My name is Saha. I am from Guwahati, Assam. I am currently pursuing my M.Sc. Chemistry from Borooah College Autonomous, and today I will present the assignment on molecular docking and lead discovery. And the given protein to me is tau protein, which is related to Alzheimer disease. So here we will discuss the Alzheimer disease shortly. This is a progressive neurodegenerative disorder, leads to memory loss or cognitive decline. Two main abnormal proteins are responsible for this. One is amyloid beta and another one is tau protein. So here I have attached the two figures which I have found in research papers, and one is according to the researchers that in the diseased neuron, the microtubules gets dissociated due to the tangled tau protein. So this is due to the hydrophosphorylated tau protein. By the means of hydrophosphorylated, it means a chemical modification where too many phosphate groups are attached to the protein. So uh, the tau protein, which is our target. Tau protein is microtubule associated protein, and in this figure, I have shown this is taken from software PyMol, which is 3D visualized of the structure tau. Tau mainly maintains the neuron stability, and so here in the main objective of this assignment is to identify potential tau inhibitors and perform molecular docking with it, uh, analyze different binding affinities of different ligands, and which is the best ligand suitable for it, finding it. So here I have shown all the 5 ligands I have selected and found on literature and research papers, uh, which is related to the molecular docking of tau protein. And the binding, these are the binding ligands showing different binding affinities. So I will perform molecular docking with these ligands and showing the results are in methodology. And the data I have collected is as given the format. Protein I have collected from UniProt and clean the structure by using Discovery Studio. Ligand I have collected the name of the different ligands uh, by being through different literatures and research papers. So I have collected all the ligands from the PubChem and saved it from 3D SDF format. And by using PyRx, I have done all the other methodologies for docking. So in molecular docking, using PyRx, first I have collected the I have turned the macromolecule and the ligand. This is the two figure 1. This is showing the final interaction with the ligand and the macromolecule. Here I have shown only the hesperidine ligand, which is given the best value among all of the other ligands I have done. So this is the structure of the hesperidine ligand. This is the 3D visuals from PyRx. And first, I have done the minimization of energy of the hesperidine ligand, and then run AutoDock Vina, and then we got all the result, and I have analyzed the results. So as of I have seen, among all the different ligands I have used, hesperidine is giving the best result, which is minus 7.5 kilocalorie per mole among all of them, because the other ligands I get is below minus 6, which is I think not a good docking score. So I have particularly select the hesperidine, and yes, I got the results. So this is the visual data and analysis I have shown in the next slide. So for visualization and analysis, I have used PyMol 3D visualization software, and interaction analysis, I have run the Discovery Studio. Here the two figures. In the figure number 1, the yellow dotted lines are the hydrogen bonding showing the interactions, and in the figure number 2, this is the interaction of the ligand and the tau protein, which not properly interaction, but yeah, I have found this in the Discovery Studio. But at the final stage, Vina split, I couldn't, well, unable to download the Vina split, so I can't perform the another results. So this is the least I can find. So in conclusion, we get hesperidine shows the strong binding among all the other 5 ligands I have done, and potential it is the potential tau inhibitor candidate, supports structure based drug design, and this has the scope for further future validations. And so this is what I get as conclusion, but I will try to do better in future further. So yeah, thank you.